Your body uses two different processes in order to divide your cells. The first process is called meiosis, and meiosis is when you need to make copies of sperm or eggs, otherwise known as gametes, the reproductive cells. We're not going to talk about meiosis today. Today we're going to talk about mitosis. So mitosis is production or replication of all somatic cells. So somatic cells are all the cells in your body that are not the gametes. So anything that's not a sperm or an egg. This includes skin cells, red blood cells, white blood cells, bone cells, all right, everything else. Now, how mitosis works is you start out with the original cell. This is called the parent cell. And the parent cell has the nucleus here that contains all of those chromosomes. Then what happens is when the parent cell needs to divide, the nucleus is going to replicate itself. So you need to have now two nuclei that are both identical to one another. And this is very important so that the new cell is identical to the parent. After that, the cytoplasm is going to divide in a process called cytokinesis. And then we're going to end up with two daughter cells that were identical to the original parent. So each of the two new daughter cells are going to have exactly the same chromosomes and the same number as the original cell. And this is what makes mitosis different from meiosis. In meiosis, you have variation, genetic variation, which is why children don't look identical to their parents, because the sperm and eggs have variations from each other. Whereas in mitosis, all the cells need to be identical. So for example, your skin cells are constantly dividing because your skin is shedding at all times. You need to make more copies of it. Or if you had an injury, like if you had a cut in your skin and your cells need to replace the worn out or dead ones. Also, your red blood cells are constantly recycling and new red blood cells are being formed. And your white blood cells and your muscles if you're growing muscle cells, things like that. Mitosis relies on something called the cell cycle. And the cell cycle has three main stages. The first stage is called interphase, and this is the longest stage in the cell cycle. Now, interphase is divided into three more stages. It's divided into G1, S, and G2. So the gray in this circle is interphase. The second stage is actually the mitosis stage, which is shown in this little colorful piece of the pie. And then finally, the third stage is cytokinesis, which, like I showed you before, is when the cytoplasm actually split. Let's talk first real quick about interphase. So interphase starts with the G1 or growth 1 phase. During G1, now you have a cell. You can envision maybe a skin cell or a red blood cell. First, all of the organelles need to double in number. So like the mitochondria, the ribosomes, lysosomes, things like that all need to double. And then the cell needs to accumulate material needed to make DNA. And finally, the cell needs to decide, should it divide or should it not? If it's a skin cell or a red blood cell, probably yes. If it's a neuron, neurons can't divide. You're born with all the nerve cells you'll ever have. So neurons don't pass the G1 phase. After the G1 phase, now the cells go into S phase. This is also part of interphase. S stands for synthesis because this is the stage where DNA gets synthesized. So, for example, in your cell, you originally had 46 of these chromosomes, but during this stage, it's going to double to 92 chromosomes. Finally, G2 phase, this is the last stage of interphase. This is when the cell decides, yes, we should definitely divide, and it's going to produce any proteins or enzymes that are needed for cell division. Now the cell's finished interphase, it's going to enter into mitosis. Mitosis has basically 5 steps, all right? So this image is showing you interphase, we had 46 chromosomes. After the S phase, we now have 92. So during prophase, you're going to start to see the chromosomes condensing and all of a sudden the nucleus is going to start breaking down a little bit. What's going to happen is this thing called a spindle. It almost looks like a spindle from a spider web is going to form. So by the time you get to the second stage, prometaphase, the chromosomes are now starting to line up in the center, but the nucleus has disappeared, the nucleus has dissolved, and the spindle is fully formed. And what this spindle is going to do it's going to kind of, I like to think, grab these chromosomes and it's going to line them all up into the center of the cell. And when they start lining up, this is when you're officially in metaphase. Now in anaphase, the spindle, those spider web things like sticky spider web, is going to pull the chromosomes apart. So half the chromosomes are going to go to one side, the other half to the other. But remember, each half is getting an identical set of chromosomes. Finally, we enter telophase, the final step. Now the spindles are going to disappear. You're going to see each half of the cell is going to have an identical copy of chromosomes. Now the cell is going to start to kind of stretch apart here in the middle, and eventually it's going to pull apart during cytokinesis. So that's the steps of mitosis. It spells PMAT. Prophase, prometaphase, metaphase, anaphase, telophase. So once again, overall, the steps of the cell cycle are interphase, mitosis, and cytokinesis. As this picture is showing you, interphase is in the orange. It is by far the longest phase, which means your cell spends the majority of its time in interphase, whether it be G1, S, or G2. Mitosis and cytokinesis. Mitosis is shown here in the lighter blue, and cytokinesis here in the darker blue. The cell spends very little time in these stages. Hey, are you thinking what I'm thinking? This picture sure does make me want to have some pie. Speaking of pie, what's your favorite type? Apple, blueberry, pumpkin? Hey, email me your favorite type of pie, and I'll give you an extra bonus point on exam 2. Don't forget, this is highly top secret, so only those who are watching this video should email me this secret code word. Now, your cells aren't just constantly circling in these stages. They have to be really controlled. Your cell's normally waiting to enter G1. So there's something called the cell cycle control system, which controls whether or not your cell is going to divide or it's not going to divide. So what I mean by this is, let's say you have a cell, and you can just envision it being a skin cell, just so you can visualize it as something. And now normally your skin is replenishing itself at a consistent rate. So every 12 hours or so, your skin cell will divide, and then another 12 hours goes by and these guys divide. What would happen if your skin cell suddenly started replicating out of control, and maybe instead of every 12 hours, it was replicating every 4 hours, right? You would have this uncontrolled skin production, and cells would just start piling on top of each other because cells are not dying fast enough and these new cells are being made and they're piling up. So that could lead to something called a tumor, which is bad. You don't want that. And this would be basically uncontrolled cell growth if cells are growing too fast. Or you could have the opposite effect where maybe a skin cell is dividing too slow. Instead of dividing every 12 hours, the cells are dividing every 48 hours. That would mean your skin's not healing itself. It's not replicating enough. That would be a problem too. So the cell cycle needs to be very controlled. It needs to replicate cells using the proper time. It needs to not replicate them too fast or too slow. But even more important is that the cell needs to make sure there's no major mutations in the skin cell. Like, what if this parent skin cell had an extra chromosome? Instead of having 46 chromosomes, that had 47, or even less chromosomes, that had 45, and then it started replicating, and then these cells would become mutated. The cell needs to really make sure everything is good, everything's looking good, like a checkpoint to make sure that there's no problems. Otherwise, this is how cancer is formed, right? And this skin cell example I gave, it's about 12 hours for a skin cell to divide, but every cell is different. So for example, your neurons, they never divide. Embryonic cells may divide every 2 hours instead of every 12 hours. Red blood cells may divide every 20 hours. So every cell in the body is different. This was just an example. So in the cell cycle, there's 3 checkpoints to kind of make sure everything's going okay. So I always like to start the cell cycle. I mean, it's a cycle, so there's really no starting point, but in my brain, it starts here at G1. This is when you have a fresh, brand new cell, and during G1, it's going to start thinking about dividing. It's going to start preparing some enzymes potentially for DNA replication during the S phase. And it's going to start replicating some cell components like mitochondria and ribosomes and things like that. And although all the checkpoints are important, I think the G1 checkpoint, in my opinion, is the most important, because this is where it all begins. If a mutated, damaged cell accidentally makes it past G1, it's almost like going to be really hard for it to not replicate at that point. So this is, in my opinion, the most important checkpoint. So during the G1 checkpoint, the cell needs to make sure that there's no major DNA damage. It needs to make sure that enzymes are prepared for DNA synthesis, and it needs to make sure there's enough cellular components and organelles in the cell to proceed with cell division. If everything looks good, we get the green light and the cells can continue on into S phase. If there's a huge problem, something does not look right, then the cell can enter something called G0. And this only occurs during G1. So it's either go after G1 or go into G0. In G0, the first thing the cell is going to do is try to repair. It's kind of like if you had a car and it's not working, it's broken down, you're going to bring it to the car shop to try to repair it. So that's your G0, the car shop. So first, the cell is going to try to repair itself. If it can repair, it can leave G0, go back into G1, and proceed onto S. But sometimes it doesn't get repaired. Like, haven't you guys had that car that just kept having problems and you kept putting money into it and nothing you could do would repair that car? So what did you have to do with that car? You had to get rid of it. So the same thing with the cell. If the cell can't repair, then it might do something called apoptosis, which is basically a programmed cell death. And basically what that means is the cell will destroy itself so that it doesn't replicate. Sometimes a cell is in G0 for so long, it's just trying to repair itself so long that it kind of gets stuck there forever. It doesn't undergo apoptosis, but it never actually gets repaired. It kind of just gets stuck in this permanent G0. So for example, that's what happens with the neurons. They enter this G0 state and they never come out of it. They're just stuck there forever. So that's the first checkpoint, the G1 checkpoint. Now, if the cell passes the G1 checkpoint, it's going to go into S phase. Remember, during S phase, it's going to replicate the chromosomes, replicate all the chromosomes. And then it's going to proceed to G2. And remember, in G2, this is kind of the place where the cell is going to prepare for mitosis. Enzymes are going to be made to begin mitosis. Now, at the end of G2, before mitosis starts, that's where our second checkpoint is. So the second checkpoint is the G2 checkpoint. Now, the G2 checkpoint is checking to make sure the DNA replicated properly. So what that means is, let's say you had the cell, it's getting ready to go to mitosis, and you have 46 chromosomes, but during the S phase, all the chromosomes got replicated. So now there's a copy of all the chromosomes. But let's say one of the chromosomes didn't replicate itself, or multiple chromosomes didn't replicate itself. This would be a problem because this would mean that the new offspring wouldn't have the proper number of chromosomes. So that's what G2 is checking for. It's checking to make sure that the DNA all replicated properly. Again, if it did replicate properly, everything looks good, we're going to go on to the next phase. But if it didn't replicate properly, then the cell is going to have to try again to repair itself. And if it can't repair, it's just too damaged, then apoptosis. The final checkpoint is in mitosis. It's the mitotic checkpoint, or the M checkpoint. This checkpoint happens between metaphase and anaphase. So remember, in metaphase, all the chromosomes are going to line up in the center of the cell. Then you have these spindle, the microtubules, that are going to connect them. And remember, then in anaphase, the next step, the chromosomes are going to get pulled apart. So what's happening in the M checkpoint is they want to make sure the chromosomes lined up perfectly in the center. If some of the chromosomes didn't line up, right? They didn't line up properly, or they weren't attached at the spindle, that would be a problem because that would mean the new cells wouldn't get the proper number of chromosomes. So once again, M checkpoint is checking to make sure that the new daughter cells will have the appropriate amount of DNA. If there's something wrong, they need to try to repair it. If they can't repair it, the cell will undergo apoptosis. So you can see these checkpoints are so important for ensuring that the somatic cells don't have these major mutations that will spread to the new daughter cells.